ladies and gentlemen, and welcome to the Nixon Library. My name is Jim Byron. I'm the president and CEO of the Richard Nixon Foundation. Welcome to day 2 of the inaugural Nixon National Cancer Conference. Last night, we looked back at the history of the National Cancer Act, and this morning, we will get into its practical uses and implications today. I want to begin by thanking a few individuals here with us, starting with our conference chair, Dr. Andy von Eschenbach, former director of the National Cancer Institute and commissioner of the FDA. Andy spent 3 decades as a physician and is now the president of Samaritan Health Initiatives and a professor at MD Anderson Cancer Center. Thank you, Andy, for all that you've done. We also have 15 directors of NCI-designated cancer centers or director designees from those centers here with us who've come from across the country, and I want to thank them all for being here as well. We also have several members of the board of directors of the Richard Nixon Foundation: Melanie Eisenhower, a child life specialist at Children's Hospital Philadelphia, and the youngest granddaughter of President and Mrs. Nixon. Thank you for being here. The chairman of the board, Dr. Jim Kavanaugh, thank you for being here, Jim. Gavin Herbert, founder and former chairman and president of Allergan, chairman of Regenesis Bioremediation Products, and namesake of the Gavin Herbert Eye Institute at UCI. Thank you, Gavin, for being here. Maureen Drown-Nunn, a teacher, passionate advocate for education, and consummate volunteer, former television host who has served on our board for a very long time. Where's Maureen? There she is. Thank you for being here. And Charlie Zhang, a serial entrepreneur and restaurateur whose generosity made possible all of the technological innovations in this very room that we are taking advantage of today. Thank you for being here, Charlie. I also want to recognize Mike Elzey, director of the Nixon Library from the National Archives, who I believe is here somewhere with us. Thank you, Mike. To introduce our conference chairman and set up our first panel of Nobel laureates, it's very much my pleasure to introduce Marlene Malik, who helped immensely to bring this conference and the special exhibition that I hope you'll all have a chance to see to fruition. Marlene received a presidential appointment from President George H.W. Bush to the National Cancer Advisory Board, where she served 6 years, and then co-founded Friends of Cancer Research with her friend, Dr. Ellen Siegel. Friends, as the organization is known, is an advocacy organization based in Washington, D.C. that drives collaboration among partners from every healthcare sector to power advances in science, policy, and regulation that speed life-saving treatments to patients. She is now vice chair of Friends and was with Ellen Siegel from day one. She's on the boards of the MD Anderson Cancer Center and Duke University Cancer Center and is chair of Marymount University in Virginia. She also played a vital role in both conceiving of this conference and bringing it here to the Nixon Library, because about 2 years ago, Hugh Hewitt invited Marlene to dinner, and they hatched this plan. Clearly, cancer research is near and dear to Marlene's heart. Would you please join me in welcoming Marlene Malik?
Thank you so much. Well, good morning, and welcome to the Nixon Library. We're gathered here today, this morning, to commemorate transformative advances in medical research, treatment, and care. And this was brought about by the National Cancer Act over 50 years ago. Spearheaded by the Nixon administration and President Nixon personally, Democrats and Republicans came together in 1971 to transform medical research and treatment. And that's why I can't think of a better location for today's conference. And it's my fondest hope that we will come away from this conference with a renewed appreciation for what humanity can accomplish when we dedicate resources and commit to a common goal. So let's begin the day with a discussion of how the National Cancer Act changed from observing manifestations of cancer and employing empiric interventions for treatment to one of understanding the mechanisms of cancer, and employing rational, targeted interventions. So it's important to start our conference in this way to understand the most amazing transitions. Rational medicine based on understanding of mechanisms is now the reality for all acute and chronic diseases, and may even pave the way in the not too distant future for regenerative medicine, in which we as a society not only eliminate disease, but restore health. So to lead this discussion, it is my pleasure to introduce our conference chairman, Dr. Andy von Eschenbach. Andy has poured his heart and soul, and most importantly, his mind, into the planning of this conference. And we thank you, Andy. Andy has the rare distinction, there's nobody better to have spearheaded this important 50th anniversary. Andy has the rare distinction of having served as both National Cancer Institute Director and Commissioner of the FDA. He was President-elect of the American Cancer Society when President George W. Bush nominated him to lead the NCI. So Andy has spent 3 decades as a physician and now the president of Samaritan Health Initiatives. And he's also now a professor at MD Anderson Cancer Center. So in 2006, Time magazine named him one of the 100 most influential people who will shape the world. So ladies and gentlemen, please welcome Dr. von Eschenbach.
David? Well, good morning. Before I introduce my panel, let's probably take a little bit more of a historical perspective as to how we arrived at this point in time. Last night we looked at some of the history behind the formation of the National Cancer Act, but you know, it goes back a little bit further than that. If we go back 100 years or so to the turn of the 20th century, the preoccupation of science was to understand the fundamental nature of matter and of energy. And Nobel laureates, brilliant scientists, were probing the mysteries of the atom and its nucleus. And as they understood those mysteries, they unleashed forces that changed the course of civilization. We got everything from atomic energy to quantum mechanics to material sciences like lasers, and even an understanding of that little thing running around the nucleus called the electron, so you're all strapped to cell phones and iPads, etc. If we fast forward, because of some of that progress, around the turn of mid-portion of the century with the discovery of DNA, science's quest began to move from understanding the fundamental nature of matter and energy to understanding the fundamental nature of life. And brilliant scientists, like those we're going to meet in just a few moments, began to probe the secrets of the cell and its nucleus. And as those secrets have been unraveled, once again we're changing the course of civilization by being able to take control of life processes. Now, the way in which that was able to come about is because scientists focused on the most egregious disorder of life, the most egregious disorder of normal life processes of growth and differentiation, and that disorder was cancer. And so when we think about the impact of the National Cancer Act and what it mobilized, it enabled and mobilized brilliant basic scientists like the 3 you're just going to meet to use their skill, talent to probe the cell and its nucleus, to probe the cancer cell and its nucleus. And they have been unraveling the secrets of the genetic, molecular, and cellular mechanisms that drive our life processes. It's really a privilege among all of those scientists who have been involved in this quest to introduce three of them to you this morning to share their perspective on their journey and the impact of the National Cancer Institute. Dr. David Baltimore, who's with me here on the stage. I love the idea that he started close to Philadelphia at Swarthmore, but of course moved on to Rockefeller Institute, MIT, and was president of California Institute of Technology. Received his Nobel Prize in 1975 as he was probing the mysteries of reverse transcriptase and how RNA and DNA interact. We have with us virtually Dr. Phillip Sharp. Phil got his Ph.D. at the University of Illinois, was at Caltech, Cold Spring Harbor, and has now been an integral part of MIT. He was probing the fact that DNA had segments that did not, in fact, translate into gene expression, and began to understand the diversity of how from some genes we get so many proteins, and how that coding of RNA has now led us to many other ventures that you'll hear about. But he received his Nobel Prize for that work in 1993. Jim Allison, fellow Texan, got his PhD at the University of Texas, was at Scripps here in La Jolla in California, back to MD Anderson, Sloan Kettering, and then now back to MD Anderson, where he's been unraveling the mysteries of the immune system and how that has altered and changed our lives in profound ways, for which he received the Nobel Prize in 2018. We're privileged to have the three of them to share their journey and to share their insights. And I'm going to begin by asking them each briefly to comment on this evolution and revolution of science that they've been a part of, and particularly how they think the National Cancer Act of 1971 and the impact that that's had may have contributed to or accelerated that process. So I'm going to begin, David, if you will, with our virtual partners. And Phil, Dr. Sharp, if you'll begin by telling us a little bit about how you think the National Cancer Act has been impactful in this evolution of science.
Well, it's had a large number of effects and really tremendous effects. But I want to speak a little bit about the basic science side of cancer, how we understand the nucleus and the cell, and how the National Cancer Act impacted that. Because in the National Cancer Act, there was the acknowledgement of the need to get a greater understanding of the human cell. And that led to a number of basic cancer centers. And MIT was one of those centers, and that's where I have spent my career. But more importantly, in 1971, I left Caltech, and after completing a postdoctoral stay, and was looking for opportunities to do science, particularly focused on human cells, particularly focused on the problem related to cancer. And because of the act and the expansion of Cold Spring Harbor with the support of a National Cancer Program project grant, I was able to find a position with Jim Watson, of Watson and Crick fame at Cold Spring Harbor, and therefore launched my career into studying how viruses interact with cells to cause cancer, because we could manipulate viruses at that stage. And it gave us a tool to probe these processes in a very specific way. And then a few years later, MIT established the Basic Cancer Center under the guidance of Salvador Luria, another Nobel Prize winner, a great friend of and leader in science. And that led to my recruitment to MIT down the hall from Dave Baltimore, which was a major inducement for my accepting that position. And I have seen this blossoming of our knowledge of the cell due to the contributions of enormous community. But that community was nucleated and supported by the leadership and the resources and the national effort to have impact on the lives of patients by advancing our understanding of cancer and treatment of cancer and control of cancer. And that has been an enormous success story.
David, you started your attention to DNA, and we're looking today at being able to do genetic engineering and CRISPR, et cetera. From your perspective, what was that trajectory like, and what was the impact of having the National Cancer Act and the NCI?
Well, as opposed to Phil, I started working in virology in 1960, 61, first as a student at MIT and then at Rockefeller. And I did that. I chose to work on viruses because they were the simplest form of life. And I thought we could make advances that took advantage of the knowledge of the centrality of nucleic acid and the structure of nucleic acid, which had been elucidated by Watson and Crick in 53. And it was a good choice because it was the only organism, if you call a virus an organism, that was simple enough to manipulate, that you could begin to understand its fundamental nature. By the end of that decade, the 1960s, I realized that the only thing we had left to understand in virology was how a virus caused cancer, because the rest of it was going to happen. It's still happening, as we all know. And so I set out to understand RNA containing tumor viruses because RNA was clearly the central player in the transition of information from DNA to RNA to protein. But we really didn't understand how an RNA virus could exist, that could cause cancer, that could permanently change the way a cell grew. And luckily I found the path to showing that the virus particle itself had an enzyme that could reverse the flow of information from RNA back to DNA, and therefore prepare a molecule that could stably modify the behavior of a cell. And so we at that point suspected, although I must say we didn't know yet, that the way that these viruses control cells is by having genes that could be reverse transcribed into DNA, put into cells, and control the growth of the cell after that. And that was all before the National Cancer Act. In fact, I've always sort of wondered what impact that work had on the passing of the National Cancer Act, because it was well known that we had done that. And of course, the major figure in that whole story that's not here to tell his part of it is Howard Temin, who was a great scientist and had for 10 years been saying that this was how things were going to play out. Once the National Cancer Act was passed, there was a national commitment to taking this node of knowledge that we had and expanding it out. And there was a recognition, and I must say, to me, that recognition was embodied in Benno Schmidt, who was the head of the National Cancer Panel, I guess we call it. And because he said to me, he said, this money is not going to go down the paths that we've been investigating cancer traditionally. It's going to go to fundamental work so that we can understand from the bottom up what it means to be a cancer cell and what it means to drive a cancer cell to continual division. And that's how it worked.
Brilliant. And we'll come back to some of that because there really is some interesting background history. But Jim Allison, you know, what, more than a century ago, Paget came up with a theory that the outcome of cancer, the outcome of a disease, is the interplay between seed and soil. It was the seed and soil hypothesis. So it wasn't just the cancer cell, but the environment, the micro, macro environment the cancer cell finds itself in. You've devoted your career to understanding that micro, macro environment from the point of view of the immune system. How has the National Cancer Act transformed immunology using tumor as a model system, and how has that influenced your career?
Well, thank you. It's been a terrifically big influence, actually. I was a graduate student at UT Austin when the Cancer Act was passed. I was studying biochemistry, but I got involved in some tumor projects and did some experiments that led me to show something that had been shown in other labs, but that we could cure mice of cancer with chemotherapy, basically, and then re-challenge them. And they were forever immune to re-challenge. And this really, at the time, really caught my eye and interested me. And thought, and about that time, T cells were discovered, and about all that was known was there were these white blood cells that go through your body, not just through the blood and the lymph, but percolate through the tissues and look for viruses that are infecting or whatever. And I immediately thought, well, maybe they can detect cancer too. And even though that wasn't what I was studying, but I switched into immunology for a postdoc and then got a job at MD Anderson, which I think was made possible in this about 1975 by funds from the NCI. MD Anderson was one of the original 3 cancer centers. And I had a project that was more directly related to cancer in an indirect way. But I started my work there trying to understand how T cells worked and worked out the structure of the T cell antigen receptor, which was the first key, and just have been doing that ever since. And went to Berkeley, did some work showing. And by the way, my work was funded, even though it didn't really, on the surface of it, have anything to do with cancer at all. One of my early grants was from the National Institute of Allergy and Immunology, but then I began getting funding from grants from the NCI, extramural grants from the NCI, and that funded my work all the way through the discovery of inhibitory pathways in T cells, and the notion that if you can block these inhibitory signals, which serve one purpose is to protect normal cells, you can unleash the immune system to treat cancer. And after we had the fundamental work, I felt that I needed at Berkeley. And again, that was funded, that work was still funded, even though I was not at a cancer center, it was still NCI funding. But then I went to Sloan Kettering to really, try to usher this work into the clinic and learn about the clinic myself, and ended up returning to MD Anderson about 8 years ago to try to expand it. And was lucky enough to take this just fundamental research that I was doing, and what regulates the immune system, and every now and then venturing out from that fundamental work to do a cancer experiment, learning something, and then going back to the fundamental biology, venturing out. I still consider myself a basic immunologist. But the bottom line is that most of the clinical work that we started doing was in melanoma. When we started this work, and when the drug that we developed, ipilimumab, was approved in 2011, the median survival after diagnosis of metastatic melanoma was 7 months, with fewer than 3% of patients alive at 5 years. I was telling Phil just before we started, the report came out this week with a 6.5-year follow-up of a phase 3 trial of my drug plus a second generation drug. It blocks another molecule, where the 6.5-year survival is 58%. So 58% of melanoma patients are now alive 6.5 years after they were treated. And so we're trying to, what we're doing now is trying to extend that to other cancers, and it's been successful in many other types of cancers, not quite that well. And so we've got a ways to go. And some cancers like glioblastoma and pancreatic have not responded yet, but we've got a ways to go. But I think as we go back and combine now what we've learned with regulating the immune system with some of the more classical ways of treating cancer in a way that we don't hurt the immune system, I think we're going to be able to get those response rates, and not just response rates, but I can consider them cures. I think about 20% of people that get monotherapy with ipilimumab are alive 10 years plus after treatment. So when I say 6.5 years, that's just following it. It's been there for a long time. It's likely to stay there for 10 years or more. I mean, these patients basically don't have to be looking over their shoulders anymore and worrying about recurrence of that disease. But with more work, I think that we're going to manage to do better. We've got a long way to go, but it's a start.
These are great testimonies to the critical importance of basic research and the need to continue to foster that. You can't solve a problem you don't understand. And you're right, Dave. Prior to the National Cancer Act, Congress had allocated $250,000 for a study on cancer that came forth as what has been called the Arbor Report. And it said that, based on work you were alluding to and others, that the science was emerging and the timing was right for the commitment, so that if we could understand cancer, we then may be able to do something about it. And a lot of what you've alluded to, and Phil brought this out, a lot of this occurred in the context of this interplay between basic research and clinical research and the observation of the clinical realities. I had a conversation with Josh Letterberg at one point, who started out as a medical student, decided then he didn't want to be a physician, he wanted to be a basic researcher, scientist, but he said he doesn't think he would have ever gotten his Nobel Prize if he hadn't those clinical perspectives and insights. You mentioned, Phil, the relationship with the Cancer Center, and one of the big parts of the National Cancer Act was immediately funding 15 more cancer centers, and now we have 71 plus, et cetera. Talk a little bit about that interplay between basic research and the clinical reality and the clinical research, and particularly from the point of view of your exposure to cancer centers. How has that made a difference? Phil, you want to start with that?
It's made an enormous difference. And it's made an enormous difference at various levels. So, for example, in the mid-70s, when I made the discovery of split genes, that, as you mentioned earlier in the introduction, the gene comes in pieces and is spliced together to make the functional protein. Right down the hall between David and my lab was Bob Weinberg, who was struggling to isolate a gene that he had identified in human cancers that showed tumor type activity in cells and culture. And understanding the gene structure and the chromatin around it and other features of basic science, he was able to isolate the first human mutant gene, a RAS gene, from a cancer, and show that cancer is a genetic disease. But all of that depended on having clinical cells, tumor cells, in an environment in which this interest of bringing genes to understanding the basic science of cancer was essential. But then as a cancer center, it participates in the National Cancer Institute processes. As a director of the Cancer Center at MIT for 8 years, I became a director in the mid-'80s. You participate in policy-making issues, you participate in review committees, you participate in meetings sponsored by the National Cancer Institute, you participate in bringing people together to form grants, and you participate in sharing of knowledge with everyone, all the way from the most clinical people to people who are more directly involved in the interface between clinical and the basic cell and the cell activities. And that community, which is beyond the university, it's beyond the lab, it's a larger national and international community, brings progress very rapidly from one site across the country, and it instills a vision of people working together to really impact on the lives of patients. And so when an innovation such as the immune blocking therapy that Jim was beginning to develop, that has become a part of the national cancer research community, and trying, as Jim just described, to find ways of expanding the activity of this immunoblocking therapy to other types of cancer. But it's a very dynamic community, bringing young people in, training them, providing them resources around the country, and then developing with this focus on how to better understand and treat cancer. But I will say, it also has allowed us to see a lot of other diseases in terms of new understandings of cell interactions and others that are critical for all medical care. So the National Cancer Act not only impacted on cancer, it impacted across all biomedical science and biomedical treatment now, and was the seed in large part for the whole biotech community that is now very strong in this country.
We'll come back. We will come back to that. But Jim, I know you personally, from the point of view of both when you were at Sloan Kettering and now at MD Anderson, you love hanging out with clinicians like Chris Logothetis, et cetera. How has that interplay between you as a basic scientist and clinicians who are dealing with the reality of cancer in the clinic, how's that influenced or impacted your hypothesis generation, your trajectory of research?
Well, in several ways. I mean, one of them is just learning from them the sense of urgency about the need that patients with lethal kinds of cancer, need help, and how we need to, move our work along as quickly as we can. But also, particularly when I moved to Sloan Kettering, I learned about some of the hurdles you have to get through. Just showing something in a tissue culture, showing something in an animal model does not necessarily, mean it's going to work. You have to be able to go that extra distance to go into humans, because there is no perfect animal model for the human immune system or anything else. So you've got to actually try it in people, and you've got to realize that you're dealing with things that could actually harm the patient as well. And there's a balance between those two. Moving the drug fast enough to help people, but not doing it so fast that you become reckless and causing a lot of harm. And I just also learned that you can also, from a number of clinicians, including Chris, but Pam Sharma, about the value of doing trials in small numbers of patients and analyzing them much in the way we would a mouse experiment, dissecting down to the cellular and molecular level what the impact of whatever the treatment we did was, so that even if you don't hit a good home run and see a clinical outcome, you can see from learning enough about the signature that's associated with a good outcome, if you try something new, are you on the right path? And if you add two things together that give you individual components, you can begin to do a lot more good and have a lot more effective therapies than if you just try a single one. And you can learn a little bit about that in the laboratory in mice, but there's no substitute what I've learned from doing clinical trials, not big ones where you're looking for a statistically significant difference in survival or anything like that, but just data seeking trials where you treat patients with amounts of drug where there's a chance of helping them, but you get biopsies and really study. You try to learn from every patient. I've learned that if you do a trial where you don't learn something from every patient, you've really wasted a bunch of patients and wasted time and really done harm, I think. So that's the main thing I've learned is think very hardly about what you're looking for, and then go in and get the tissue and the blood and look at the changes and try to see the impact so that you can learn the next time, are you blocking this particular pathway and not another one? And how do you put all these together? Because that's how we're going to move from now on. I mean, melanoma is an example of two blockers together, but it's going to be about a lot more than that, combining, as I mentioned earlier, more conventional therapies like chemotherapies and tyrosine kinase inhibitors in ways that can prime immune responses to get us over the top. And anyway, clinical trials are obviously an essential part of that.
Well, it's a beautiful model of, we used to think of bench to bedside, but what I hear you're describing and what you've alluded to is it's now a circular process that we start with discovery, development, and when we get delivery, the delivery reinforms us in terms of some of the basic discovery that needs to occur. And David, you were kind enough with others, Nobel laureates and basic science, come to be to NCI and meet with me and help inform the agenda. And I remember at one session, you made the comment that there's so much more we need to learn about cancer, but there's also the opportunity to use what we already know in a more strategic way to intervene in those critical steps in the process, whether it's metastasis or initiation, et cetera. Speak a little bit more about your continued effort to go beyond your own work to really drive that strategic approach to dealing with the clinical reality of cancer by understanding these fundamental mechanisms.
Well, I'm a basic scientist and I love working on basic mechanisms. And I think it's important that there be a cadre of people who actually don't respond to the urgency of the cancer patient and who drive knowledge deeper and deeper into the nature of the normal cell, the processes in the normal cell, and how those get bastardized in a way in cancer cells. But I also have always thought it important that we test our knowledge by trying to apply it. Because it may be too early. It may be that we don't know enough. It may be that we don't have the right context for treatment. But it may be that we do know enough. It may be that, at least for some cancers, and I think Jim Allison's focus on melanoma is really interesting in that regard, because that was the cancer that was, in a sense, easiest to handle immunologically and most responsive, and where you could make the greatest difference. But there are still cancers for which we simply don't have an approach. And again, Jim mentioned pancreatic and glioblastoma, but there are plenty of others. In fact, there are melanomas that are still killing people. So we don't have a complete solution to any cancerous problems. And so we have to go back to the basic science and say, well, there's something we're missing. What is it? And often we don't know. We have no idea. I mean, if I go back to 1960 when I started in basic science, we literally had no idea what was going on in a cancer cell. We didn't know about oncogenes. We didn't know about suppressor genes. We didn't know about reverse transcription. We didn't know about anything. We've had to learn all that. The amazing thing is that in those 60 years, we've learned all of this. We've gotten to the point where for some patients, we can say, we have the materials to allow you to live a normal lifespan. But for so many others, we don't. And we have to keep the focus on basic knowledge. I will point out that you have 3 people on this panel, all of whom are PhDs, none of whom have an MD, none of whom are actually trained in medicine. And that shows you where the knowledge comes from that allows an advance in something as totally challenging as the cancer problem.
Yeah, well, you were 3 amazing seeds. We just planted you in the right soil, which was the cancer center. You've alluded to something I want to touch upon and get all 3 of you to just converse about this. The progress that's been made in cancer and our understanding of cancer and understanding some of these fundamental underlying mechanisms is now applicable across all diseases, acute and chronic. We can see that things are now cancer-led, not cancer-centric, and that science is becoming, if you will, horizontal across these. And it's a little difficult, I don't want to go too far with this, but we've been trapped in the old model of observing manifestations of disease phenotypically, a lump in a woman's breast, a shadow on an x-ray, and we designed and organized accordingly. But now we're seeing that when we get down below the macroscopic and microscopic to the molecular level, there are similarities that transcend diseases. We're looking at SARS-CoV-2 right now and mRNA vaccines, et cetera. Talk a little bit about your perspective on how our understanding of cancer is now impacting our ability to understand and manage a whole host of other diseases. Jim, you're in tumor immunology, but yet we have immunology that's affecting autoimmune disease. We have infectious disease that's if all of you could share with us and converse with each other as to where you think we are in the integration of science and where it could lead us using cancer as that discovery system. Jim, I pointed you out. You want to start with how you see
I'll say that very early in our studies, when I realized we wanted to get a really aggressive immune system response to cancer. And so one of the things we did was studied autoimmunity to try to understand how it worked and ways of making it worse, to tell you the truth, with the reasoning that if we could do that, and you do the same thing in the context of cancer. But now we're trying to go the other way and figure out how to use those same lessons to turn it off, the ones we were trying to shut off before, and making some progress there, not enough yet, but some drugs have come out of some of our cancer work that are now in use in treating some kinds of autoimmune disease. But one of the things that we begin to realize lately is that things are not quite as simple as we think they are, because a postdoc in my lab has some very early information, but very intriguing data showing the neurotransmitters and neurological sensors and products of nerve cells that can influence the activity of T cells within a tumor. And we haven't linked that up to the nervous system as larger thing, but I think that it's going to lead us into perhaps interaction of the immune system with the nervous system, in both in causing neurological diseases, but perhaps also in neurological regulation of immune responses in some cases. So it's just speculation at this point. I don't know. We've just got the early molecular fingerprints that suggest this is going on.
And moving us into areas of neurodegeneration and Alzheimer's, et cetera. Phil, you're talking about RNA, and we have mRNA vaccines. What do you see as the horizontal dispersion of what we've learned in cancer to other diseases?
It's been absolutely enormous. I was involved in starting Biogen and a biotech company, a large biotech company, the oldest freestanding biotech company in the country. In 1978, we saw the interest in interferons becoming a treatment for cancer and others. But let's look at the COVID-SARS-2 situation. We now have RNA vaccines where we can redesign a vaccine in 2 months and show it's safe in patients and move it into treatment. This has really changed how we are responding to this challenge. And if you look at our understanding of the virus and how the virus has evolved, and how our immune system and our monoclonal antibodies, which we now make to treat this virus, how it can evade that immune response. All that information is a product of this lateral engagement of breakthroughs. But I just want to mock something David said. This venture is not even close to being over. We're seeing an integration of engineering and high throughput computation and large databases into our understanding of cancer, how to predict who's going to develop cancer, how to find it early, and how to then intervene more effectively. So we are not close to the end of the journey of cancer. We will be making enormous progress over the next 20, 30 years, all the time improving the outcome of the lives of patients.
David.
Well, as Phil just said, the impact of the knowledge that was generated through the National Cancer Act and through the focus on cancer as a problem has permeated the way we deal with every disease. When you think about what disease is, a large part of it is infectious disease, and that has certainly been an area of the application of the knowledge that's come out of cancer. But a lot of it's genetic disease, and there's no question that the way we approach cancer, a genetic disease, has influenced the way we approach all inherited disease, inherited at the level of the transmission through generations, as well as inherited as a consequence of mutations in the body. And we're now using genetics as a probe for almost all diseases because there is a genetic component, there's an inherited component in the way we respond to diseases, the way we respond to infectious diseases as well as others. So it's all mixed up together. And I think it's worth spending just a moment thinking about training, because new generations come up every decade, let us say, to take over from us who have done our part. Where are they trained? They're trained in basic research laboratories. MD PhDs are getting PhDs because they need the approach that is taken in a basic research laboratory. And then it's possible to move into disease-focused institutions and to bring institutions that have a basic source, like the MIT Cancer Center or Caltech or the Broad Institute, to take a more recent addition, to focus on the disease elements that we don't understand and that we can illuminate by basic investigation.
That's a perfect lead-in. So the idea of the young mind and training. So here's what we're going to do. We're going to make the three of you 50 years younger. I'm going to take away your Nobel Prizes. And you're now reflecting on this 50 years of progress, so you're starting right now. What would you want to study right now? Where would you focus your career on the mystery that you would like to solve, given the 50 years of progress, given the fact that we have stem cell biology, we have computational biology, we're moving from an era of experimental biology to theoretical biology, et cetera. You're now at 2021, 50 years younger, and you don't yet have your Nobel Prize. Well, what are you going to do? You're all smiling, but nobody's answering.
I'll tell you what interests me lately is something that we've found in others. It's not unique to us, but just a surprising mutability, plasticity of cells. We used to think there's given states, and we have textbooks that says you've got this state, this state, this state, this state. And after they differentiate, they're immutable. And we're finding that's just not true. Immune cells in particular have the ability to take on a lot of the characteristics of the cells around them and to respond to signals and change their complete transcription patterns and even look like different kinds of cells. And it's just understanding that, the mechanisms that are involved in that is something I think I am too old to do it now, but I think if I was a little bit younger, that's what I would start thinking about.
So I will follow up on that, because if I was starting now, I'd try and approach the thought process this way. But life science is informational science. It's informational that has been with us for 3 billion years as organisms have defined. And now we see our tools to understand the nature of that information in a cell, and the whole explosion of IT, machine learning, large capacity computing. And I think that integration over the decades in front of us is going to reveal some remarkable, deep understanding of both cancer and all of life science. And I would try to be in the middle of that mix if I was young and doing it again.
There are two ways to think about what you're going to do at the outset of your career. One is to sort of follow your nose. A guy like Ed Wilson cared about ants, and he went and tried to find every ant in his community and to give it a name and to understand its properties, and he became a central figure in ecological science. But others of us, like I was interested in viruses. It doesn't matter where you start. It matters what you do. And that is to really investigate at a fundamental level what's going on in whatever it is you care about. The other way to decide what to do is to kind of look globally at science and say, where are the big open questions? Evolution is a big open question. But the brain is a big open question. And that's one that will benefit from all the things Phil was just talking about and lots else, because it is so complex, involving, you know, numbers that I can't even remember how many zeros are involved in them, skillions and trillions and whatever of synapses. So I mean, there's no question that that's a challenge to anybody who is looking for a way to go into science and to find a big question. Of course, there's dark energy and dark matter, and you could go after that. But I don't even understand the words, never mind the way to go after it.
Well, if we can keep some of your progress going with stem cells, we'll get you those 50 years back pretty easily. You know, we're in the midst of a pandemic of infectious disease nature. Alzheimer's is in many ways a pandemic affecting us globally. And in 1971, cancer was a pandemic. There was not one family that was not touched by this disease. The work that you, the three of you and your colleagues in basic research have helped us come to grips with being able to solve that pandemic. There's more work yet to be done, but we know how far we've come. You've opened the doors for us to be able to solve the pandemic of today with SARS-CoV-2, and you'll help us solve the pandemic of tomorrow, neurodegenerative diseases. So we are grateful to you not only for your participation on this celebration of the 50th anniversary of the National Cancer Act, but grateful to you as basic scientists and your colleagues for helping us understand and deal with a disease that was virtually a death sentence 50 years ago when this all began. Thank you. God bless you for your work. And we're looking forward to the next 50 years of your contributions. Thank you.